Chapter 8. Once we're through with Chapter 8, we're through with the unit. So the cells unit includes Chapters 6, 7, and 8. And there's 8.1, 8.3, and then we have 2 more strips after that, 8.4 and 8.5. So we're almost through with the cells unit. Whoa. I'm not a good gluer. So 8.1 has a lot to do with energy and how the cell has to maintain function when there's not a constant input of energy. Also, it deals with some of the other pieces that we learn with thermodynamics about order and disorder. So the second law of thermodynamics, okay, so here we go with the second law, is all about order and disorder. And this is particularly important in biological systems because being able to live within the constraints of these thermodynamics laws, in other words, just the way the world works, you've got the ability as an organism or a cell to let those work for you. So you're working within what will naturally happen. So that was very vague, but let's explain what that means. So second law of thermodynamics is about order and disorder, which is entropy. Explain the diffusion of a substance across a membrane. So giving myself context for what I mean here, so I know what I'm explaining. See, this is a lot of times the problem. They don't give you this anymore. Like, they don't just throw out second law of thermodynamics and expect that you know what that is. They have a limit as far as, like, here's what you're supposed to know. You're supposed to know there are laws, and they're Newton's laws. You've been exposed to the conservation. This is entropy. But they would tell you this was entropy. So you wouldn't have to just memorize that and know it. Explaining the diffusion of a substance across a membrane, being able to connect how those two relate is the whole point of the question. But you've got to remember, okay, when they say diffusion of a substance across a membrane, and it's diffusion, they're wanting me to talk about why do substances naturally move from high to low concentrations, and talk about entropy when you do that. So that's the way to reword that. So let's remember about entropy. Entropy is a measure of randomness. So we've talked about this before when I told you, okay, picture your room, don't pick it up for an entire month. It's going to move toward disorder. That's the natural tendency of systems, is to move to disorder. So let's write that. So there is, in other words, this is the way that the world works, the universe. There is a natural tendency to move toward disorder. Okay, now I've talked myself into a connecting point here, because if there's a natural tendency of things to move toward disorder, and there's a natural tendency for substances to move from high to low concentration. If I put those two together, it tells me that moving from high to low concentration is moving toward disorder, because it's both the natural process of the way things work. So let me think about why that is. If I showed you, let's go back here, this picture, and I said, hey, what do you notice about the hydrogen ions locations across these two places? So we had said this would be like a cell membrane. This might be where you've got the blood vessel running next to it. But you have two different compartments. What do you notice about the hydrogen ions? What you should be able to say is, and I'll just add some more here to be sure we're overly dramatic. I notice that there's more on this side of the membrane than on this side. Could that randomly happen? Probably not. And we actually learned that, I was about to say yesterday because that's when we made the video, but I don't know when you're actually watching them, that we did not naturally create that. We purposefully created that by pumping hydrogen ions so that there would be more on this side, less on this side of the membrane, which is a gradient. So gradients do not naturally happen. Let's write that, our next statement. So let's make some just thoughts. First of all, gradients are purposefully created. And remembering what a gradient is that you have, so just remembering that a gradient is more of something in one area and less of the same thing and they're going to say in an adjacent area, just because we need to be able to move from one to the other. So you remember when I was a moth and we had light by the TV, and away from the TV it was darker. So we had a gradient of light. Here we have a gradient of hydrogen ions. More of something adjacent to less of something. Those don't naturally happen. They're created anytime that there is an action that created something. In other words, it didn't randomly happen. That's where you're going to reduce entropy. It's bringing order. So if I order the system here, I am pushing these hydrogen ions to where that the order of this system is more here, less here. So it took energy to do that. There was work involved, just like there would have been energy and work involved for you to pick your room up. And you ordered your system. So let's put that there. So when gradients are created, that right there gives you that understanding that it's intentional. Order is created. And I think I'm going to change that to just order is brought to the system. And the reason you would think about it is because there is work or energy used to create that difference, that high and that low. So that high concentration versus the low concentration, right? So that was purposeful. So think about it this way. Your room's a hot mess, and you're going to bring order to your socks. That's where you're going to start. So you go in your room, all on the floor, under the bed, on top of the bed, on top of everything, in the basket, in your bag, everywhere in that system, and you collect your socks and you place them in the drawer. So there's a compartment that's adjacent into the bedroom where there's a higher concentration of socks. So now let entropy happen. You just don't order the system anymore. What will happen is there'll be a natural flow of the socks out of the drawer. You'll get the socks, you'll wear them, you'll throw them on the floor. Pretty soon, there'll be this movement from putting the socks in the drawer, which created order, which reduced entropy, to letting natural processes happen, which the socks go on your feet, which then get thrown on the floor. They're not ordered anymore. It's random. So when you have diffusion and it's moving from high to low, this will occur until there's dynamic equilibrium, so that there's this equal concentration across this barrier, whatever it was that was separating the compartments, and then you'll have just dynamic equilibrium, which means whatever number of molecules cross to one side, the other same number across to the other side. So the concentration has been equal. So it will naturally fall down its concentration gradient because things move toward disorder. So we'll put that. So we'll put systems, which that would be what this is, a compartment with high adjacent to a compartment with low. Systems naturally move to randomness, which is disorder. So molecules following this natural pattern will naturally move from where you had purposefully ordered them in the high concentration to where that it's a random amount. You've got the same concentration on both sides, but you have dynamic equilibrium, and those molecules could be found in either compartment with equal probability, which is random. So they will naturally move from high to low, and we'll just put on here to make this answer the question, because that is going to increase entropy, which is disorder or lack of order. I hope that makes sense. That's an excellent question. There's so much content knowledge that you have to bring in here in order to answer that. So that's the big focus of 8.1. 8.1 has a lot of just Gibbs free energy and the reason why that you have different reactions that will occur spontaneously or that you have to add energy to occur. So there's a lot in that that we really don't go over a whole lot. But this particular part right here, there's a lot of good content knowledge that you have to bring here. So we're going to skip over to the 8.3 and talk about energy coupling reactions and their value to organisms. So I think what I'll do is I'll just number this 8.1, make this one 8.3 number 1. So let's think about this and what they're actually saying. What do they mean by energy coupling reactions? That's the part I've got to figure out first before I can even think about why that's valuable. So what they mean is, let's just do some words. And you know all this. The terminology will be a little different because in chemistry, you're going to talk about exothermic because there's a release of heat energy. But in living things, that's the thing we don't want. Last thing you want is to have reactions work and release heat energy. Think what would happen to you. Like literally you would blow up in heat and light. So biological systems don't use those words. So we're going to write some reactions release energy. You know that. This is called, for us, instead of exothermic, it's exergonic. So important and a buzzword. Those are the ones that release energy. Then there's some reactions that you have to bring energy to for them to occur. So now I'm going to write that. Some reactions require energy. Those are called, instead of endothermic, they're endergonic. Exergonic releases energy. Endergonic, ender, like you're going to put into, they have to have energy to move forward. So here's the thing about living systems. You have to have the ability to manage your energy. So the best way, whenever you're trying to like work in your head, okay, I'm trying to get a good feel for the way that energy is working in this system. Honestly, the best analogy and the way to think about, okay, everybody's got experience with money. So whenever you are struggling and thinking about energy, step out of the biology world, move into your world, and think money. Because energy is the currency of the cell. So I'm going to stop answering this question, and I'm just going to describe a situation for you. You work, everybody at some point has done some job for money. You may not have like a job where that you work for someone else, but we've all done something where we did something for money. So we're going to step into the adult world, and trust me, it happens faster than you think, where that your job is linked to your pay. So I work, I'm paid, but that money that I'm paid, I'm just going to pretend it's like it was when I first started teaching, which this was a nightmare. I was paid one time a month. So as soon as I was paid, that money wasn't all spent the second that it went in my bank account. I had to have a way to conserve it, save it, so that I could use it in little pieces over time to do the work that I needed to do. So I was paid, I don't know, let's just go with something simple, $1,000. From that $1,000, the second it went into my bank account, I didn't have to immediately spend it all because I didn't have a way to keep it. It didn't dissolve or dissipate just because I received it. I had to capture it in my bank account, hold it there, and couple it with a process to pay my mortgage, pay my car payment, pay for food, pay for electricity, pay for water. Those type of different processes that I needed the money to do the work for. Exactly what they're saying here. So energy coupling means I have reactions that require energy. Where am I going to get that energy from? I had different processes that required money at times when I was not getting paid. How was I able to have the ability to spend the money when I needed it, when I wasn't always paid at the exact moment that I needed to use it? Well, I had a bank account, right? That was the coupling for me. So what's a way to conserve this energy so that I can spend it later? So let's write that. What is a way for living things, cells, to capture this energy that's released, and I'm putting in all of my buzzwords here, from exergonic reactions, so that energy isn't lost. I mean, it would be really sad if I worked on teaching and the second that the pay was given to me, that $1,000 we decided we were going to use to make things simple, if I didn't immediately use it the second that it was available, that I lost it. It was gone. And yes, that would be awful. But in this particular case, when you have reactions that release energy, just like I was being paid, I didn't blow up if I didn't spend it and it was lost. But when you lose energy in a system, it is lost as heat and light. So we'll put here. Well, neither one of those can do work. Awful. So what do? Well, what did I do? I had a bank account that captured that energy, that paycheck, and held it available to be used in any amount that I wanted. So it wasn't like, again, I'm making this up, that my $1,000 paycheck was this amount that was held together and you used it all or you didn't use any. So if I had a mortgage of $300, I had to waste the $1,000 because it came as a big chunk of currency. I need like pocket money. I need to be able to pull out $10 to go out to eat or $300 for the mortgage, not this whole chunk that was made at one time. So I also have to conserve it in spendable amounts. So what the cell does is it's going to use ATP as the spendable amounts, pocket money. Now, I told you that my cute grandmom, which I don't know if I ever told you, she was 4'10" and 72 pounds. Like, that's my grandmother. Can you imagine? Her feet were tiny. She wore a size 4 shoe. So she could literally put on Hunter's little shoes. I can still see her walking around in the shoes he had that lit up. And she was the cutest thing. She's tiny, tiny. But she would call this walking around money. So what you were doing is you were having money that was in a spendable, so let's put this here. So this would be energy in a small enough amount to be usable. I don't want to go and have to make a decision. All I really wanted was a Coke. it's $1. Do I really want to take my chunk of money that is all in one thing of $1,000 to like waste it for $1 for a Coke? Of course not. So you have to have it, your energy, in spendable amounts. ATP is the perfect spendable amount. It's the currency of the cell. So what happens is this. You have an exergonic reaction. It's going to, which we said already, release energy. And instead of blowing up in heat and light, because I can't couple it to something that I'm going to use that energy for, it's lost, living things store that energy in ATP. So you use this energy that would have been lost, and it becomes held in an energy holding, capturing molecule that I can spend whenever I need it. Well, when would I need it? Well, I'm going to use ATP because ATP is going to be able to provide energy because I've captured it for endergonic reactions that require energy, like a bank, right? So I made money, a chunk of money. I don't think I'll probably spend it as a chunk. Oh, that's okay. We'll put it in the bank, and if you need $1, or if you need $100, or if you need all 1,000, you can have it. We have it held for you in spendable amounts. Oh, thank goodness. Because I need some, because this process requires energy. I'm going on vacation, and that requires money. Okay, go to the bank and get your money. That's all this is. So when they ask you to explain energy coupling reactions and their value, holy cow, isn't that easy to know what the value is now? The value is you don't lose that energy every time you have a reaction that releases it. Living systems would not survive. So the value is that the energy generated by these exergonic reactions is available to do work. And I'm just going to put because it is stored in ATP. Thank goodness for not blowing up in heat and light, to do work, which would be the work would have been to provide energy for some type of reaction that requires it. Well, let's learn a little bit more about ATP then, because dang, that's pretty awesome. So you're going to label ATP, which is down here. We've already got cards where we know what these are, and describe how it performs its work. Well, I'm pretty sure that this question is about structure leads to function. How this molecule is made is going to be the reason why it does the job it's done. So let's go through the structure. Let me see, here we go, with these sort cards, if I can find, so that right there, looky there, nitrogenous ring, that's a nitrogenous base. What I'm looking for, hey, that's what I'm looking for right there. I didn't do that on purpose, but I think that's awesome. We learned this was called a nucleoside because it's not quite a nucleotide yet. Nucleotides are the monomer, the basic subunit of nucleic acids. It's a sugar the phosphate's what we're missing, and a nitrogenous base. So ATP is this nucleoside with 3 phosphate groups on it. So you're going to have a sugar, a nitrogenous base, and 3 phosphates. So let's go over here. So let's go on the other side of this. So this one, I'm going to keep it out. And I'm going to go back over here, and we'll just leave it sitting here so we can think about that. So this is number 2. So I'm supposed to label ATP. Even though they've got a drawing over here, I'm going to be sure that I can draw it myself. So there are 2 pocket monies, 2 different energy currency, molecules in the cell. And I'm just going to put in living things. The first one is the ATP. The second one is GTP. They're exactly the same, except in ATP you have, look here, it's adenosine, because that's the word for adenine, plus this ribose nucleoside. You have also a different 2-ringed purine, right, nitrogenous base called guanine, and so that's what you would have in GTP. So when they say for me to label, I'm going to draw. I'm going to look at this. I'm not worried about that double ring, putting in the double bonds and all of that. I'm just going to allow myself to know that I've got this purine. In this case, it can be adenine, where did it go, or guanine. Those are the only 2. It is going to be covalently bonded to a 5-carbon sugar. So this is the purine. This is your nitrogenous base. It's bound to ribose, which is a 5-carbon sugar. This is a nucleoside. And then the actual energy collecting, storing part comes in the 3 phosphate groups that are attached to this sugar. So if you've ever played around with magnets, which I know you have, and you put like sides together, you know how that they fight you. Like, depending on how strong the magnet is, you literally might not be able to put them together. These phosphate groups have a charge. So just remembering, I don't think I have my functional groups cards in this notebook. But if you looked at them and you went back to your functional groups cards and you looked at phosphate, because as a functional group, they're what gives the function of the molecule. So phosphate groups are negatively charged. They do not easily get stuck together. So, bonding 3 phosphates in a row would require quite a bit of bond energy. So if we think about it, if we had a MP, which is adenosine monophosphate, adding that phosphate to this nucleoside, it's not pushing against, fighting against, charge-wise, anything. It would be a really low energy bond. You add the next phosphate group with this bond, now you've got that repulsion to where you're forcing two negatively charged groups, these phosphate groups, next to each other. This bond has more energy than this bond. So this would be ADP, diphosphate, and then this last one would have the most energy because now you've got these repulsive factors here, and this one is being added into that. So this bond right here has the most energy. They usually call that a high energy bond, and a lot of times they'll use something that looks almost like is equivalent to type of sign. This would have less energy, this bond that would take you from ADP to AMP, and then this bond here that holds the one phosphate doesn't have enough energy really to do anything with. So the way that ATP performs its work, which is right here, that's what the question is. So I labeled it. I've described its properties. The properties come from the functional group. The functional group is this phosphate group. How ATP performs its work is the energy that you have from exergonic reactions provides the energy for an enzyme to put this phosphate group on. So I'm going to have to go to another page. Think about what that is. So if you have a MT, which is adenosine, which is what we call that nucleoside that's adenine plus ribose, mono 1 phosphate, and you have an enzyme that's going to add what they call a PI, which is just a phosphate group that's free. This actually is called inorganic, which means I'm not attached to a carbon-based molecule. Phosphate, it's just a loose phosphate group. So the enzyme adds this phosphate to A and P. So it's going to get its energy, so because a bond is energy. So the energy for this bond that you're making will come from an exergonic reaction. So we capture that released energy into a bond, and we go from AMP plus a phosphate to ADP, which is now the same nucleoside, adenosine, which is adenine plus a ribose. Now it's diphosphate, and it's holding energy in that bond that's between the two phosphates. This bond right here. So we didn't have to have any type of reaction that had a lot of energy, because that energy in that bond is lower than it is going to be in the next one. So now you'll have an enzyme that adds this third PI to ADP. Energy comes from some exergonic reaction that we're not going to lose that energy, definitely not as heat and light. That's dangerous. So we'll capture that energy, and use it to make the bond, and you'll end up with ATP, which is adenosine, which is adenine, right, plus ribose. That's what this is. That's a nucleoside, and it's tri, because there's now 3 phosphate groups. Phosphate. So this will be plus 3 phosphates. So then as you need to use it, so this is the way we make it, you're going to take ATP, and then you'll have some enzyme that will remove, so it's going to remove, and the enzyme removes this phosphate. Now it's not inorganic anymore because it's bound to this whole molecule, removes the phosphate at the end. Energy is going to be released, so we'll release energy. Now most of the time it's used to do work. It may cause a protein to change its shape, like those cool motor proteins that you saw walking. See, this is me walking along the cytoskeleton. Every kick that it throws that foot out costs an ATP. So it's almost like a cattle prod. That energy, when you stick that phosphate on there, you phosphorylate that protein. It's like a cattle prod with whoop, and then it whoop, and so it's able to walk. It might be that. It might be that you use ATP to drive another reaction, but you've got that energy, you're spending it, so you release the energy to do work from releasing that phosphate group, and so you end up with ADP plus that phosphate now is back free into the system to be used again up here and up here. You still have more energy in that molecule, you just don't have the amount in that second break, so you can, instead of just letting the ATP be regenerated into ATP, which is the most common thing that cells do. If they're low on energy, they'll take that ADP, they'll release that energy that's in there in that second phosphate bond to do work. So you've got some enzyme that will remove that phosphate now that's at the end again, the second one, and you'll end up with a MP plus a phosphate. So when I think about this, I remember when I used to, like, when I had money and I would give it to Hunter. Let's describe his wallet. So in his wallet, there's his wallet, he would have his spending money, which would be whatever the cash was that he could use for whatever. And then in this pocket right here, we had a special bill. It was an emergency bill. So if you think about ATP, the energy that's released to form ADP as the spendable cash, this is the normal thing. This is what we do all the time. Then when the wallet is empty, over here ADP, I put more cash in it. So I made ATP back by putting energy back into the molecule. And we went back and forth between full wallet, empty wallet, full wallet, empty wallet, ATP, ADP, ATP, ADP. Well, where do you finally get AMP? When, holy cow, empty wallet, there's no cash to put in it. What am I going to do? Well, I have an emergency bill that I only get when I cannot get the money out of the wallet, and I use it when I'm desperate. Cells rarely move by taking this phosphate off of ADP to go to AMP. In fact, creating AMP is a signal to cells that we're in trouble. Because just like with Hunter, I've spent my cash. I don't have any more cash coming in. I'm going to have to spend my emergency money. I'm in serious trouble. So this molecule is a signal to let the cells know it's in an energy deficit. Okay? So I feel like we've answered all those questions. This actually is, I think, a test question that was maybe, I don't know, like part A of a long free response or something, where they wanted you to give what ATP is, like you could draw it or describe it, and then describe how it works, which is always structure to function. Okay, so probably won't have a lot of videos with this because it's just the way it is, plus thinking about my budget. But I hope that makes sense, and if you have questions, you email me. You're the best. I haven't said that in a while. You're wonderful.
In the enzyme-catalyzed reactions tutorial, we talked about the thermodynamics and kinetics of chemical processes. We saw that Gibbs free energy, or G, tells us about the stability of the starting materials and the products. The change in free energy, delta G, is used to determine the spontaneity of a reaction, whether the reaction would, if possible, proceed in a given direction. We contrasted this with kinetics, which tells us about the rate of a reaction. The activation energy is the energy barrier that must be overcome if a reaction is to proceed, and it determines the rate of reaction. Activation energy is used to determine how likely and how quickly that process can happen under given conditions. And we saw that enzymes can lower activation energy by binding and distorting molecules, bringing them together in just the right orientation so that the reaction occurs. In this tutorial, we'll look at adenosine triphosphate, ATP, the cell's energy currency. We'll apply these principles of thermodynamics and kinetics to ATP, and we'll see how ATP can be used to drive other non-spontaneous reactions. Here's the structure of ATP. Notice that adenosine is a ribose sugar covalently bonded to adenine, a nitrogenous base. Adenosine is used not only in RNA, but also as a form of energy storage when covalently bonded to 3 phosphate groups. Each phosphate group has multiple negatively charged oxygen atoms. As we saw in the DNA and RNA tutorial, these negative charges exist because of electron transfer between hydrogen and oxygen, forming O- and H+. The hydrogen ions dissociated into the watery environment of the cell. Just like magnets, like charges repel each other. As a result, phosphate groups that are covalently bonded together store potential energy. It's as if you took the repelling ends of 2 magnets and tied them together with a string. Because your string is strong, the magnets don't fly apart. But if you were to cut the string, that potential energy would turn into kinetic energy, and the magnets would move away from each other. The covalent bond between phosphate groups is like this string. It's strong enough that the phosphate groups stay together, but if it were broken, the phosphate groups would separate and energy would be released. Not only does breaking this bond separate those repulsive negative charges, it also increases entropy. While the phosphate group and the rest of ATP must be in the same place if they're bonded together, once that bond is broken, the phosphate group can be randomly located near or far from adenosine diphosphate, or ADP. Additionally, water molecules with their partially positive hydrogen atoms now have better access to the negatively charged oxygen atoms of both phosphate and ADP. All of these things create a large driving force to break apart ATP. Here's the reaction. ATP is broken into ADP and a phosphate group called inorganic phosphate, or PI. This is a hydrolysis reaction, where a molecule of water breaks the bond between the phosphate groups. The hydrogen ion that is made dissociates into the cell and is sometimes attached to ADP, or inorganic phosphate, so often it is not written in the equation. Energy is released when ATP is broken, which means that the reaction is spontaneous in this direction and delta G is negative. ADP and inorganic phosphate have lower free energy than do ATP and water, and that's because ATP is storing potential energy by bringing those phosphate groups close together. Remember that Gibbs free energy tells us about the energy of a molecule that can be used to do work. Our cells use the energy of ATP hydrolysis, breaking ATP into ADP and inorganic phosphate, in order to drive many non-spontaneous processes. The energy released by breaking off a phosphate group can be immediately harnessed for other purposes. This is through coupling reactions, which requires the help of an enzyme. In the enzyme catalyzed reactions tutorial, we looked at the first step of glycolysis, the phosphorylation of glucose. We saw that overall this process is favorable because G6P and ADP have lower free energy than do glucose and ATP. There are really two reactions happening simultaneously in this step, breaking ATP into ADP and inorganic phosphate, and joining glucose and inorganic phosphate to form glucose 6-phosphate. We know that the delta G of this top reaction is negative and the reaction is favorable. The delta G of adding a phosphate group to glucose is positive, and this reaction is unfavorable. Phosphate wouldn't naturally form a covalent bond with glucose. These molecules are more stable separated than they are joined. This is a non-spontaneous reaction and it requires energy input. That energy comes from ATP hydrolysis. As an analogy for coupled reactions, imagine this scenario. You're standing on a trampoline. Your friend is standing on a deck above that trampoline. Your friend has gravitational potential energy. If she jumps, she would naturally fall toward the trampoline. As she falls, her potential energy is converted into kinetic energy. Once she hits the trampoline, some of that kinetic energy is transferred to the trampoline, which changes conformation. The movement of the trampoline is then transferred to you, who gain kinetic energy as well as gravitational potential energy as you fly into the air. Your friend is ATP, which loses potential energy as it's hydrolyzed into ADP and inorganic phosphate. Much of that energy is transferred to hexokinase, the enzyme which is the trampoline. As hexokinase undergoes a conformational change, it provides the energy to drive the phosphorylation of glucose, which in this analogy is you flying into the air, a non-spontaneous process. Conveniently, we can find the free energy of a coupled reaction by adding the free energies of the component reactions. For the first step of glycolysis, the hydrolysis of ATP is more spontaneous than the phosphorylation of glucose is non-spontaneous. In other words, the delta G of the overall reaction is still very negative. Under normal cell conditions, the coupled reaction is spontaneous and virtually irreversible. ATP is used in a similar way to drive many cell processes. The potential energy stored in the bonds between phosphates is harnessed to do work. But if the hydrolysis of ATP is so favorable, why doesn't ATP break apart on its own in cells? Remember that how easily or quickly a reaction takes place has more to do with its activation energy than with delta G. The activation energy of ATP hydrolysis is high enough that ATP does not hydrolyze without the help of an enzyme. This is what allows us to use ATP as energy storage. With all these ways to use ATP, there must be some way to regenerate it. This is the purpose of cell metabolism. Regeneration of ATP is the reverse of hydrolysis. It's forming a covalent bond between ADP and inorganic phosphate to result in ATP. Remember that the delta G of a reverse reaction has the same magnitude but the opposite sign as the delta G of a forward reaction. To form ATP, not only must any energy barrier be overcome, but enough energy is required to store potential energy in the bond between phosphates. This reverse process, the synthesis of ATP, is also achieved through coupled reactions, using the food we eat as the energy source. The covalent bonds between molecules of food store energy. Cellular metabolism is the process in which those bonds are broken, releasing energy that can be harnessed to form ATP.